Hi, thanks for joining me on this episode of the Leukopak Lady. In cell therapy, we have a long history of considering cell number as the primary marker of leukopak quality. Today, I want to explain why that mindset is increasingly misleading. And as I mentioned in a previous episode, cell counts have been the focus of leukopak quality metrics because they were easy to measure, they were familiar from blood banking and bone marrow transplant fields, and they were convenient targets for teams to track. And while quantity still matters, it no longer tells the whole story of leukopak quality. Relying on cell count targets alone can mislead cell therapy development programs because the immune cells in the bag may have poor functional fitness. Functional fitness refers to how cells actually behave in the manufacturing process and in the patient. Cells with good functional fitness have the ability to expand in culture, survive processing and cryopreservation, respond to and kill targets, and resist exhaustion and dysfunction. Two leukopaks can have the same cell counts but very different functional fitness. And having more cells cannot compensate for less capable cells. This means that cell count alone is not a reliable indicator of performance. And functional fitness should therefore become a leading metric when selecting leukopak donors. But donor fitness cannot be assumed. It must be demonstrated with data. That data should guide how collection centers screen and manage donors and how developers decide which donors to use in manufacturing. In practice, this means a donor with slightly lower cell counts but better functional fitness may be far more valuable than a donor who simply produces a large leukopak. Incorporating functional fitness testing influences the kind of decisions developers make. Defining how well will these cells perform in our process can influence how many donors to recruit overall, how manufacturing processes are designed, and how to think about repeat donors. If functional fitness becomes a leading quality metric, collection centers also need to evolve their approach to donors. Instead of simply recruiting as many eligible donors as possible, centers begin to identify donors whose cells could perform well in manufacturing and prioritize repeat donors whose cells consistently perform well in a developer's process. This would involve inviting certain donors back more regularly, tracking their performance over time, and treating them as long-term partners rather than one-time donors. It also means that centers need strong donor stewardship practices. If a developer identifies certain donors as especially valuable for their process, centers should work with those donors to support habits that maintain immune health, such as sleep, stress management, and overall wellness. This is how collection centers move from being logistics providers to biological partners. Functional fitness turns donors from interchangeable suppliers into differentiated biological assets. As you can see, donor fitness now matters as much as, and maybe more than, cellular quantity. And this is also where donor stratification comes in. So join me on the next episode for this discussion.